DNA replication. What is DNA replication? So we can call it the DNA synthesis, or DNA replication, or DNA copying, or making a new DNA. So whatever you call, it's the same thing. First, going to the terminology. Replication is the name for the process. So this is the process of making a new DNA. From starting, we have a DNA, and then we have two DNAs. This is called replication, and you can call it DNA synthesis as well. Polymerase. This is the enzyme responsible for making the newly synthesized DNAs, and the actual synthesis is done by the polymerase itself. And the polymerase, you can understand from here that polymerase, so it's a polymer making enzyme. And the other terminology is double strand. All of the DNA we are dealing in this situation are double-stranded, and these are the first strand and the other strand, and we draw it like this, but it has a more into the complication of it inside the chemical structure. We will see very soon. The DNA is always a double-stranded DNA. If it's a linear DNA, this is called a linear DNA. Or it can be circular, which I draw it like this. And this is what we see in the bacteria. And you can also draw it like this, like double-stranded DNA. And this ones we can draw like this. Whenever we draw DNA in a linear or a simplified version, in this one we state that there's a circular helix, double helix going on, but like this, now we only simplify it to show the reaction itself. The DNA is never standing like this. It's not ergonomic, or it's not optimized. It's not an optimized structure, and we can draw things like this. These are the hydrogen bonding between the bases, and this structure in here is called sugar phosphate backbone. Similarly, in this situation, we have the hydrogen bonding inside the double strands, and this is the sugar phosphate backbone, and these are the hydrogen bonding. This is a circular DNA. And there is a direction to the DNA. When you write 5' in here and 3' in here, we have to write 3' and 5'. They are anti-parallel, meaning that if this is going this direction, then this is going the other direction. And then we have the base pairing, and base pairing is a simple thing, very, very simple. Adenine pairs with thymine, and guanine pairs with cytosine. And this is always the case, and there are no exceptions to this rule, like guanine cannot pair with adenine or the other versions. It's strictly forbidden. There's a simple explanation why it can't happen. If you think about this as a circular structure, the chemical structure, then we have two rings, or ring structure or circular structure. The adenine and guanine are called the purines, and they have two circular structures within them. And thymine and cytosine are one ring structure. And whenever they pair, like adenine pairing with thymine, it occupies a 3-ring structure. And the same with the guanine and cytosine. What doesn't happen is this. For example, if you want to pair guanine with adenine, then there will be 4 ring structures, and this will be larger. And whenever you pair thymine with cytosine, there will be 2 ring structures, and this will be narrow. This will be narrower than the actual structure. And how polymerase acts is, polymerase does not go and add. Suppose that this is a DNA strand and this is the polymerase now. Does not like add one nucleotide and then go away, and then add another nucleotide and then go away. It doesn't work like that. The polymerase works like this. This is the DNA strand and this is the polymerase. It binds to the DNA structure and scans and like add another nucleotide, and this polymerase has a structure in here, like a space in here, and whenever there is a correct nucleotide binding, it fills up this space, like the 3 ring structure fills up this space. If it's narrow or larger, then it doesn't fit in here, and then polymerase does not continue the reaction. So first the nucleotide, like adenine to thymine pairing is done, and then the polymerase makes the synthesis, like, and then another synthesis, and it goes like that. So this polymerase structure is actually a huge structure. It's like, it occupies like on the DNA, and it has lots of other parts, like 10 other proteins are working in the polymerase structure, and they all make sure that the pairing is made correctly. So it evolved like this, and this is true for all the living things like eukaryotes, bacteria, viruses, all of them. The pairing is done in the same way, and this is the 2 hydrogen bonding and 3 hydrogen bonding in between the DNA strands. And another crucial thing, like, you can separate these strands by breaking the hydrogen bonding, because hydrogen bonding is a weaker bonding, and you can separate the 2 strands like just by sheer force, like opening a zipper, but you cannot break the sugar phosphate backbone, because the bonding in here is the covalent bonding, and it is a very strong bonding, and it's a very structured bonding, and you cannot like separate it. Of course you can break the DNA by enzymes. There are special enzymes, but during the replication you do not destroy the sugar phosphate backbone, you just separate the 2 strands. Like if you're opening this double-stranded helix, you just separate the hydrogen bonding, and it's getting cold in here. So this is exactly how the DNA replication takes place. You can think of this as the polymerase, and you can think of like existing DNA strands as opening up the helix, and then suppose that this is a newly made strand. Now you are making a connection in between the 2 strands together. You can open this up and close this up, make a new synthesis, but you can never like destroy this the backbone in here. That the backbone, the clothing here doesn't change. It's the same as the DNA synthesis, and whenever if you cut this zipper from here, it becomes non-functional. It is the same as the DNA. If you damage the sugar phosphate backbone, then it's a huge mistake, and the cell alarms itself, and then it destroys that DNA or kills the cell itself. So for the DNA replication, we have to start from the structure of the DNA, and whenever you look at the structure of the DNA, it looks like this, and if it is confusing for you, we can simplify this diagram. Like this part is the phosphate group, we can write it like this, and this part is the sugar group, can write it like this. And here we have the base, nitrogenous base, or simply the base. And this is the nucleotide, the three of them together. And this is the base, which is making hydrogen bonding, and the double-stranded DNA, the double strand, is following in here. And then we have another phosphate group and another sugar group, and then the base again. So the sugar phosphate backbone is here, and the bases are only inside of the double strand. If we want to draw the double stranded sequence, it will be something like this: base, phosphate, sugar, base, and then here we have the bases. I'm drawing the other strand, and this have to be connected to sugar. So you can start drawing like this, and this will be on sugar again. But now, because they are anti-parallel strands, you have to draw it the reverse side. So the phosphates will be here, and then phosphate, and then sugar. So this is how to simplify the DNA, and this is how it looks like chemically inside the backbone. Now for the DNA replication, we have to consider the directionality of the DNA, and the DNA has a direction which is like 5' to 3'. And where does it come from? It comes from the sugar. We always remember the sugar. This is actually like this. Let me correct it. It's coming like this, and then like this, and this OH is connected in here. So in this structure, 1, 2, 3, 4, 5 carbons. If you have problems with enumerating the structures, I can draw it in here. So when we draw something like this, there is an oxygen here, but there is a carbon in this position. We don't draw write the carbons for simplicity, but whenever there is an intersection of the bonds, it means it's a carbon. This is true for all the organic compounds, and if it is carbon based, obviously. Then there is another carbon here, then there is another carbon in here, and another one in here, and then the other one is coming out of it. So this is the one, first, second, third, fourth, and fifth carbon. And each carbon has to make 4 bonds. So if this is like going this way, 1, 2, 3 bonds, then it means there's an H connected in here. 1, 2, so it has to be 2 H hydrogens coming out of it. And we don't write the hydrogens as well. This is also true for all the carbon-based compounds. And this is the numbering of the carbons is like this. So because the phosphate, here is the uppermost structure in here is the phosphate group, and it is connected to the 5th carbon, so it is called the 5' end. And because this is the 3rd carbon and it ends in here, this is called the 3' end. And the most used version of this is 3' OH group and 5' phosphate group. This is crucial in understanding the DNA replication because all the polymerases that we know of work only from the 5' to 3' direction, and there is no polymerase identified which works in the opposite way. All of it, like from all the eukaryotic cells to the prokaryotes, all of them work in the 5' to 3' direction, and we will see how and why it is working like that. And because this is an anti-parallel structure, if we call this 5' phosphate group and 3' OH group, then the other strand would be 5' to 3'. If you like to simplify this more, you can just draw a double helix and call this 5' to 3' and this one 5' to 3', so that they will be in anti-parallel to each other. And then the bases will be in here, base, protected from the outside, buried inside the double helix, and the sugar phosphate backbone will be on the outside of the DNA. And whenever we draw a helix like this, it is the sugar phosphate backbone. Now, okay, we can continue with the polymerase and why it works in one direction. I will leave that in here because we need the chemical structure. So if you are struggling with the chemical structures, you can just draw phosphate sugar, but then you need to understand how structures are added to each other and why it doesn't go in the reverse direction. So when we have 5' to 3', and when we tell that it goes from 5' to 3', it means that the new coming nucleotide will be added from here, not from here, but it will be added from here. And we have here an OH group. This is why it is called deoxyribose, because in the ribose there is another OH group in here, but because this is missing, this is the deoxyribose, and it only has one OH group in here. And OH groups are usually reactive, so a chemical reaction is highly possible from the OH group because it can remove the hydrogen and make a bond through the oxygen. This is the structure of the ATP, our main energy source in our cells, and it has 3 phosphate groups, and these are the high energy bonds in between. And here we have a ribose sugar, and then there is the base, nitrogenous base. In this situation it is the adenine. But when we have the nucleotide, it is called adenosine or deoxyadenine triphosphate or deoxyadenine monophosphate, you can name it. We have the this OH group is missing, and the main difference between the energy molecule and the nucleotide that we use in the DNA is the missing OH group. So these are the phosphate groups, and whenever I use the phosphate groups, and people use this term instead, like peas with circles, these are representatives of the phosphate bonds, and it comes in here, and then it will be attached to the sugar in here, so that the phosphate and sugar backbone will be continued by addition of the new nucleotide. The polymerase, the enzyme responsible from the DNA replication, it makes bonds only in the sugar phosphate backbone. The polymerase itself is not bothered with the base pairing in here, but the base pairing makes it sure that the polymerase is in the right direction and it is carrying the correct nucleotide. So whenever there is a new nucleotide coming in from here, suppose that this is the new coming nucleotide, it has to match the corresponding nucleotide, and then whenever there is a hydrogen bonding in here is formed, the polymerase can attach this unit into the existing strand. So this pairing, base pairing, does not require any enzymes or anything. It just happens spontaneously, and adenine pairs with thymine and guanine pairs with cytosine. There's not much surprise in here, and they make hydrogen bonding in between, but the new coming nucleotide is added via the covalent bonding. So this part requires polymerase, and it requires an enzymatic reaction. So this is, I think, all we need to know about the chemical structures of the DNA. We need to look at the replication and how it is carried out. So it is called a semi-conservative replication because whenever there is a DNA to be replicated, like this, it divides itself into 2 strands, and then the new strand will be synthesized accordingly to the template. So this will serve as a template. And this is the newly synthesized DNA. Now, and then these will form the 2 cells, go into the 2 cells. This is the original cell, and these are called the daughter cells. And in each daughter cell, there is 1 strand coming from the parent and 1 strand that is newly synthesized. This is called the semi-conservative. So it conserves this one, but this is new. And it always continues like this, and it never does like this is the parental strand and this is the other strand. It never does this. Always some part of it remains from the parent. This is why it is like this, because it always requires a template. Like the DNA synthesis cannot start on its own. And it's called like de novo, starting from nowhere, like starting from zero. The DNA synthesis is never like de novo synthesis. It's always starting from a template and looking from a template. And how the template thing works, we have the DNA here, 5' to 3', and the other strand, 5' to 3', or let's write this like 3' to 5'. Each time a DNA synthesis starts, it has to be from 5' to 3', but this is only in the newly synthesized strand, not on the template, because this is the direction of the synthesis, not the direction of the template. So, and because it is anti-parallel, it starts from here, and this is the 5' to 3' on the newly synthesized strand. On this one, it starts from here to the other side, and it is called 5' to 3'. This is the synthesis reaction, and it goes in the anti-parallel strands. However, we can never like separate the DNA like this. If it's a small DNA structure, then it is possible, but in a eukaryotic cell, the chromosomes are too large, and even if it's in a bacteria, it is not possible to separate the strands like this. Instead, if we have a huge chromosome like this, 5, 3, 3, 5, it is first. So 3, 5', 5' to 3'. The synthesis takes place like this. It opens up this structure only slightly, and then because this is the 5' coming from here and 3 prime going from here. The synthesis would go like this, 5 prime, and the other one would go like this. And in the same time, simultaneously, it will go from like this and here like this. So there will be bubbles of transcription. It will just open that area and then make the synthesis, and that part will be double-stranded and separated, and the other parts, because they still have the bonding together, it will so it will follow up all through the chromosome. In the bacteria situation, because the bacteria has a circular chromosome, you can draw it like this. It opens up from here. Let me draw it like this. So it opens from here, and because it's circular, on the original chromosome you cannot find 5 prime to 3 prime direction, so it starts from somewhere. If then this strand, it goes like this, on the other strand it will go like this, and it will follow the structure whenever there is a full completed circles, 2 circles as chromosomes. And even if this is a bacteria genome, still the bacteria has lots of control over the replication. So it's not a even if it's in the bacteria, it's a well-regulated system.